Okay, so this here said is our streaming editor. Got a template file. We're gonna replace me with something, and it worked. See it right there. See it right here. See it up there. This works for anything. Echo hello world. Said search for hello, replace with goodbye, goodbye, whatever. So this is your set can do a lot of stuff, but I think its real best use is it can do search and replace. You could look for a pattern in a line and have it delete the line after that. It does all kinds of things. Not worth memorizing. Definitely worth knowing that said can find patterns and do actions based on them. And then go to GPT and say, using Bash said, here's the thing I want to do. It will give you. So that's that. This right here is really great, but I also don't want to have to do this a bunch of times. But essentially, I could cat my template, replace something, and then pipe it into a new file. Oops. Yeah, more
Where does the G?
global. So kind of like with our parameter expansion and replace, it'll only replace the first instance that it sees that in the variable name. The G here means that every time it sees it in a line, it replaces it. So if I echo that, and then pipe it said search for C, replace it with an underscore, done. I only did the first one. I'll go back and add a G for global, did it for all of them. All right, so that's said in a nutshell. And all we got to do is combine that with some sort of loop. We loop through every single one of those accession numbers, and each time through the loop, we're going to have said replace me with that accession number, and then write it to a new file named after that accession number. And suddenly we've got ourselves an arbitrary number of Slurm jobs, and we can run them all. Okay. How do we loop through that? File is data accessions.txt. I want to loop through a file. It's a bit different than we've done before. Before we did for I in some list of stuff. Now the list of stuff is sitting in a file. Let's see. For I in, how do I just cat it here?
The dollar sign.
Yeah.
Do the cat.
Echo $I done. Okay. That works.
Is that like? Third one? Is that?
Yeah, it's a third line.
That's good.
I could echo I sleep too. That'll work. But I do want to show you before the semester ends. File. There's a special one here in Bash, a while read. This is specifically for reading through a file. And it's while I'm reading through the file one line at a time, I will do some stuff until the end of the file. So while read some variable name, I do. And then what are we gonna do here? The contents of the file is this.
Echo.
Could echo I. Done.
You can give it a I.
We didn't give it a file. And this is weird. I'm gonna control C out of here. At the end, you gotta feed it a file. So here's two new things we haven't really seen: a while read loop, and then it's that little. It's a redirect, but backwards. It's saying that the file on the right side of this is going to become the standard input for the stuff here on the left. So we're actually redirecting the file kind of leftward to become the standard input for a while we read. And that works. Okay. Following so far? The syntax is weird. While reads something, some variable, do some stuff using that variable or not. Done. And then after the done, you send it to file. So if I do cat scripts 03 assemble template, there, it just catted it twice for me. It still has replace name
Why did it get it?
because there were two lines. But yeah, there's one, there's two.
This is like the while loop, the loop is just counting the number of lines and does we
It's
have
just going through each line. I didn't tell it to use any variables. So it's just going through. So I could cat this and now type it to sed like we did before. Sed quotes search for replace me, replace it with $I globally. And said, man, see if I can get this all on one line. There it is. There we go. Okay, so we're gonna read through some file. While doesn't know what file it is yet, but it's gonna read through it line by line. And each line will become the value of I one at a time. Each time through the loop, it's gonna cat our template, replace me with the current value of I. We'll do that globally. And then I feed it what it's going to loop through that file. Okay. Scroll up to where we did this. There we go. Here's the first time through. It worked. Yay. Done. Then the second time through. It worked. We're almost there. Printing it to our screen where it's useless. So what instead?
Can you make this like a new script? So you can just like do the okay or a creator and move to the script so you can put them all aside.
So into a new file?
Yeah.
So right here, sed is just going straight to our screen, like we told it to. We could redirect that to a new file, and what should that file be called?
$I assemble. That's
Sure.
Mm.
We're gonna have to do the curly braces to make sure that we can make this all one thing. Assemble.slurm. Done. If I look around, I should see them. Oh, there they are. Cool. So each of those is a separate Slurm job now, based on just that same like file that's got a list of accessions. We're kind of basing our entire pipeline on it, list of accessions. And it's the only like data that we really need to reproduce this whole thing. So that works right there. How could I automate building all these? I could turn what I just did here into a little shell script and have that be part of my pipeline. But we'll get to there in a bit. I want to make sure our pipeline actually works. The one sample. Oh, it does. Yeah.
Is there not like a way in like, I know for like in like Python, like you could just like leave like that replace me variable, like, just like as like a variable, and then just like pass that as like a parameter into the script.
Okay, so you can. That's when we're talking to Slurm.
Yeah.
We're going to talk to Slurm and say, we've got this parameter. It's going to be the values of this file.
Mhm.
Okay. But it doesn't make the Slurm scripts for you. It just wants to run it all essentially on the same array.
Okay.
And so, yes, we could go down that path, but then we kind of lose our documentation. Yeah. So I don't think it's a bad thing. It's just taking steps towards that. I like doing this because I guarantee that if you have 100 jobs you're trying to run, some will work and some won't.
Mhm.
And so then you've still got a record of separate output and error for each job, which you wouldn't have otherwise. You've got separate scripts that you can go back and look at and rerun. And so now at this point, if one of these like just doesn't finish, I can go rerun that, adding the dash continue. So there. So these make me happy, except they're not in the right place. So I'm gonna go up here and send them to scripts. There they are. So we've got our template. We use our list of accessions to make a bunch of Slurm jobs and then we submit all of them. But you're right that we're starting to lose reproducibility a little bit here. You look at my master pipeline, it's basically going in and saying, all right, get conda ready. And then here's the scripts we're going to run. Well, not exactly, is it? All of this on the pipeline, I'm just like saying, I want one day to finish all of these scripts. Never going to happen. So we're compromising here.
Is there like any like reason why like you want to batch like so many at a time? Is it not just like smarter to do like smaller batches? Because like if you have like if you know there's like a tendency for it to fail, it would just be easier to just batch a bunch of smaller jobs and just check.
Yeah, in fact, like I'm starting to not like this pipeline at all. was an idealistic attempt at the very beginning. We're gonna set up and download and do QC and assemble and annotate coverage and profile the taxonomy. Yeah, maybe for one sample, this would be a pipeline. But it's not gonna work if we have 10 or 20 samples that we want to get through. I mean, what study have you ever seen that have one sample? No, I mean, to be publishable, you're talking 50 or hundreds samples, so it's starting to like feel a bit gross to me. It's not as reproducible as I want. Can it ever be? Yeah, I think kind of is the answer. I would love to, and this, what I told you in the beginning of the semester is like, you're just gonna give somebody a pile of scripts and they can just run them. They'll take care of anything. That never actually happens. This is what Docker was invented to deal with. Is we're just gonna have a cloned computer with everything ready, and then the scripts go through it. That also breaks down if you have 100 metagenomic samples because you got to get Docker running on an HPC, at high memory, and it might take 2 weeks. No admin is ever going to give you permission to hog a node for 2 full weeks. So reproducibility starts becoming a bit more illusory. We're losing it in drips and drabs as we compromise to get things to actually run. What's our balance going to be here? You got to think about generally what I think about is the like reviewers. So I'm usually doing this to like publish a manuscript.
In terms of like the pipeline, like say like we're right now we're doing the whole process of weeding out the data that's like failing or that's not working. Like in the end, like you only include like the data that you actually use, right? So like wouldn't that pipeline be fine then? Because like after that, at that point, like, you can send it off to the reviewers and we know that all of these files like already work already.
Well, I'm never going to give them those files, right? Because that's many gigabytes of stuff. I'm just not going to go through trying to get the reviewers those files. If they care, they can start running these, and these scripts will give them the files.
Yeah.
But it's going to give them all the files we started with.
But if you like shortlist the list, like that original list, the back to like the list that you actually end up using, like the files that like aren't
bad
stuff.
Yeah. Okay, but how is that reproducible? How'd you pick these files, right? Like you picked those files that are remaining in high quality and everything, all the reads that are left and truncated based on code. So if we then go back and just start from that point, kind of black boxing the whole selection process. I'm not fishing for like the right answer here. It's like I'm complaining about it. That true reproducibility becomes kind of a dream. We're working on an HPC, and we have to work on an HPC to get this kind of job done. So I said that I think about like the reviewers. I actually don't expect them to run. I've never had one run my pipeline, but I expect them to be able to. And so the idea that I and a bunch of people I know, not everybody, goes with is kind of in an ideal world, if you could have unlimited time on a big HPC, this would work. It's documenting all the steps we took, you will never be able to run it. You're gonna have to split it up into separate jobs and do all this stuff, but here's what we did. So it's reproducible in that sense that, no, not reproducible at all. It's the word. It's fully documented, I guess. Here's all the steps we took to get to this point, but it took us all semester, so good luck doing it yourself, but if you want to, have at it. That's kind of the balance that I've gone for. Is these scripts are kind of idealized. We did end up running them all, but we had to go through a lot of trouble and like submit separate jobs to get them to run and all that. I don't know. I don't think there's a right answer, but we can do some things to make it a little nicer, at least to ourselves, like working with batch array jobs. I found that it's not actually nicer to myself in the long run because some of the jobs fail and I don't have good documentation of where and why. It's like a calculate files. Okay, all that is a bit academic. All I know is that we've got these and they're working.
Well, when you are publishing papers like this, like when you're, I'm aware of like a paper that dealt with these pipeline things.
I bet you have.
I probably did, but at the same time, do they not include like in the paper themselves, like challenges and how the researchers like what they did to overcome that in the research? Or like at what
Yeah.
point you deviate from the method you use?
Yeah, so you might see like a paragraph in the paper that says, we started with 88 accessions, and it was reduced down to 64 due to low quality. That's it. But you're never going to see this kind of stuff in the paper itself. It's just in the background for the supplemental documentation of the paper. So not in the print version. Some journals still do that. But if you're on the website, you'll see supplementary information or some supplemental methods. Like it there.
Okay.
Usually I'm just going to point people to my GitHub repository. My most recent paper been published here. This is it, the GitHub repo. Here's where you get the data. Here's the database that I used. Then here's the whole pipeline in order. Yeah, good luck getting through this one ever. This took 4 days on a 1 gig HPC node just to get that. So it's never going to happen. Reviewers are never going to actually do this, but somebody down the line might want to, because they may want to dig deeper into this particular paper that they were interested in. I'm flattering myself. Nobody's going to care about this, but like what I've done is I've made a release on Zenodo. So it's not just a GitHub, because I could always delete my GitHub repository after the fact. Here. That's right. Cool. Okay.
Oh, the one that
Oh, there we go. So this is it, the whole pipeline. version 1.1 was after review and made a couple times changes. That's it. So this is forever until the collapse of society here in a few months. So it's forever until then. I can't delete this, and it's got a DOI, so a persistent like digital object identifier, and so it's also citable. So I just go ahead and cite this in my paper. I do it right before it gets published. And I say full pipeline and methods details is available here. And that's it. And so it gets a one sentence in my paper, all this work. One sentence at the bottom in the data availability statement.
Really is at the bottom.
It is, yeah, it's bad. It's like after the acknowledgements, you know, where you thank the bartender or whatever, like, okay. So it's just living on a repository. People do use it. I get, you know, emails from grad students every month from around the world who are using bits of code and asking me like, I'm getting a bug. I'm like, not my problem. Try to help for like a day and then after that, ask your advisor, go away. But yeah, so people do use it because this is how I learned to code, was looking at other people's code. And that's how a lot of people learn to code is, you know, they've got a project they need to do, somebody else has done something similar, they're just gonna kind of like copy that code, reuse it, change some variables. So you're still providing a nice service for other people. You're providing a nice service for yourself, because you better believe I'm not gonna rewrite all this stuff every time. Really? Like a script to download sequence reader? Got it. Copy, paste. And so it's nice for that. So we've got an idealistic pipeline of scripts here. This one will have to be submitted into two separate jobs. Or for the real assignment A, it's going to be 10 separate jobs, but it's going to be that same while read. Make a template where the only thing that's changing is that accession number, and then you're good to go. So is anyone able to get that to run?
Could you could you put the assembly template? I like tried.
Oh, yeah.
But okay, I got lost. So
Oh, and you. Any part in particular?
I'm gonna try to just like rewrite it, like copy it, and then. So the replacement is in,
I put it right here in the for loop part.
yeah,
That's
so
the only important part.
so the job name, you don't need like the stars or anything, it's just the,
Yeah, because the job name, it'll just be ERR 1055 underscore assembly. I need the stars here because I don't know exactly ahead of time what that file name is going to look like, but I know it's going to have the accession.
Mhm.
And then maybe something else, like maybe there's an underscore, right? So I'm just being lazy and saying star one. QC is the quality control stuff. Has to be GC.
and then that's like the last place you need it, so it's there. You can put it
Yeah, that was the
out
last
there,
one.
and then,
All the rest
Okay.
is the same. Since our for loop does the dirty work of like deriving the reverse file names and all that based on the name of that file, as long as I have it looping through that one file. And that's what it is doing. It's kind of silly, but it is looping through it.
and then could you put the while loop back up?
Yeah.
Okay. Sorry.
These while read loops I use them all the time. They're pretty handy because I will often have like a list of gene names that I'm looking for. So I can have it read through it for each gene name, go in and like pull out stuff and move them to a separate file, and then go in and find all the. So we'll do that after like our annotation step.
Is there a reason you only use the bracketed I in certain places? In that?
It's where you're trying to use a variable and add stuff to it. So in the first place, with the set statement, we're just looking for that variable replace me. So it doesn't need to be bracketed. Right here, I'm looking for that variable replace name. Or I'm looking for ERR or whatever. But then I'm trying to tack it on to this other part of the word. So I'm making a compound word of that accession underscore assemble. If I just did $I underscore assemble, it'll look for a variable called I underscore assemble. That makes sense?
Yeah. And then at
So
the
if I,
end,
let's say I equals that echo $I underscore 123. Didn't work. It's like I don't have a variable called I underscore 123, but if I say echo I, that isolates your variable in curly brackets, underscore 123. Now it works. So it's kind of letting the $sign know that this is the entire variable name I'm looking for. All right, well, at least some of you got the assembly done. It went pretty quickly. What's it look like? Mine was in scratch 10, the assembly OA. I put it in contigs and then the name there. This is what I got as my output from spades.
Where you at? Is this one first?
Yeah, it's wherever we like sent our contigs. Did all of that basically for this file. If you remember, we're talking about like contigs or longer contiguous reads that were built out of those short ones. So there it is. We've got a whole bunch of other junk in here that might be useful for troubleshooting or like tracking down your errors, kind of seeing what happened. The entire log is stored there. But there it is, contigs.fasta. That's contigs up. Go away. Okay, I forgot, not in there. It's just listing from here. So let's look at that file. That's longer than 150 bases. Quite a bit longer. You did good. That's awesome. I've got BB map, and so that gives me BB stats. So BB stats.sh comes with BB map, and you just give it any FASTA file. And it tells you all about it. The percent of A, C, T, G, and N. IUPAC. You wanna know what those are? We'll often see DNA sequences with these other letters, like a W. That means I can't tell it's an A or a T, right? So there's a letter for each possible thing. When we make primers in the lab, if we want an A and a T at a spot, that's two separate sequences, we put a W there. TCA, TCA, AAGW. That means we've got a primer for with an A at that position and a T at that position. All right. We got 0 of those, good. Other, that's like little hyphens and things, just gaps. Main scaffold total, this is the number of contigs, essentially. 13,212 contigs.
Some different stuff on my class.
Yeah, you're good. Okay. This is a metagenome of mostly bacteria stuff from human gut. If this was a genome assembly for a human, how many contigs would we want?
They're all one, which is
Not one.
okay.
24, because one is the mitochondria, but yeah, 23 chromosomes. So if this was a single, like E. coli, we'd want one because it's a circular chromosome. But for eukaryotes, the ideal Plato's like idealistic perfect assembly would be 23 plus 1 for humans. Q.
This is less, and so it's just,
Unless you scroll through the file. Yeah, so it doesn't barf up the entire giant FASTA. And then we got stats here. Okay. And then basically kind of showing us like how big each condig is. We've got one that's over 500,000. That's a big contig. Cool. We've got 7 that are over 250,000 bases. What's good for a contig? What was the point of even making these out of our short reads?
yeah, like some actual context with the data.
Context and, I mean, how many nucleotides does it take to make a gene? Okay, depends. Depends. Is 50 enough? No. 50 is not enough. Because it takes 3 nucleotides to make a single codon. So, not gonna happen, right? So it does depend on the gene, but some genes will fit on 1,000. That's big enough for some genes. Some might take 5. But there's no gene I know of that can be encoded for in 100. Didn't we start with 150? Was our length of our short reads? Some of them are only 50 long. Did it shrink them? How does that work? Yeah, quality control. Some of them had such crappy quality at the like the tail end, like it got truncated. So yeah, we got some garbage in here. But once we start getting up to the big contigs, I usually throw out everything that's under 1,000, and that's pretty common. We're down to 2,666 for this sample. That's like reasonable. The other stuff is all kind of trash. It just didn't assemble very well. that's kind of nice. So overall, I think this is a good assembly with a bunch of garbage also, and then we would just go in and throw out the shorter stuff or leave it in because who cares? Okay, so we've got it assembled. What's next? Annotate. So the annotation script is up on our lesson as well. But it looks more or less the same. Header, setting up our directory stuff, and then we're using Proca. You got Proca, okay, it was Metafine, it's screwing up. So going in, activating our Proca environment, deriving our info for it, and then once again, it's just one line, all this work. We're giving Proca our contigs file. We're giving it an output directory, and then a prefix that'll help us like pick this sample out from others later if we merge these things. And I'm saying, hey, pay attention to bacteria. This is a metagenome. And then go ahead and add the sample name again as a tag for each gene that you find. And yeah, great. That's it. So this script isn't too different from our annotation or our assembly one. Again, all of that's just kind of set up for deriving our sample names. And then that's the line. So that's annotation. And I want to show you what the annotation looks like. Go ahead. Let me just go there. That's scratch. Patients. Boom. Okay. So that gave us a bunch of files. And here in our lesson, we've got what they all mean. But we're just going to look at a couple of them. The GFF, that's genomic flat file, I think what it stands for. And then TSV is tab separated. So it named them all after our sample, which is good. And that's because I told it to do that right here with the prefix. Let's look at the TSV. So a unique tag right here. This is gene number 1. It's a coding gene. This one's a transfer RNA. But we've got a coding gene here. This is how long it was in base pairs. So you can get a feel for like how long some genes are. Gene ID, EC number. What's that? Remember when we were going through that KEGG profile and it's like looking through it on the map of like one substrate into another one, they all had a number. That's showing what that is. And then the COG is like a kind of a gene family idea. And then the actual product, like this is what it makes. Neat. I kind of know what some of these are. Glycine transporter. I don't know what it looks like or anything, but great. I know that that's a thing. I know alcohol dehydrogenase is a thing. Glutamine fructose 6-phosphate aminotransferase. Anyone had biochem? Recognize any of those? What's that from? Glycolysis, yeah, good job. All right, so we've got a bunch of stuff in here, but mostly, what do we see? Hypothetical protein. Well, that's annoying, but they're all over the place.
Is it just they don't need the gene dose?
Yep. We don't, it was able to match it up and say, like, this is definitely a gene. Like, it's clearly got the context surrounding it in the genome. There's a start codon, stop codon. Everything makes sense within the reading frame that was there. You told me that this was bacteria. I didn't detect any like introns or anything. Like, this is a gene, but it's not in our database. Um, so they just say it's a hypothetical protein. I hate that, but it's what we've got because the way that we can annotate this is we have to know what genes are, or we can find that gene again. And so somebody may have found this before, but it has not been described in the lab because that takes a lot of work. That's like molecular bench work and stuff. You're characterizing this gene, doing a knockout of it in yeast or E. coli, kind of seeing what it does, all that stuff. So right now we've got a bunch of things that are just hypothetical proteins. So, okay, if I cat that, type it to correct B, I don't want hypothetical. All right, here's all the non-hypothetical genes. And I could then cut and take 1, 2, 3, 4, 5, 6, 7. Field 7. That's all my gene names. Type that to sort uniquely. I've got a list of all the genes that we definitely know were in this particular sample. And then I say, you know what? I don't want tRNAs. So I'm going to grep again the tRNA. Okay. Okay, so these are actual genes. They've been described before, they've been characterized. We found them again in our sample. Cool. This is starting to get at kind of a metabolic profile of this particular gut sample. We know that these genes are here. There's some other stuff too, hypothetical, that we will ignore because there's nothing else we can do with it. Or is there? Nothing that I can do with it. You as a bioinformatician, maybe not. You could take that sequence for that gene and give it to somebody who does wet lab work and say, synthesize this. Let's see what it does. We could maybe take that to AlphaFold and predict what shape that protein would be, and then see if we can classify it with other shapes. But we're still just kind of guessworking at that point. It needs to be characterized, but we're getting there. And this is a tab separated thing. You can easily import that into R or Python and start building yourself a little map of this. What we don't have now is quantification. Okay, we've got tetracycline resistance protein class B, antibiotic resistance. It was found at least once. How many times was it found? That's kind of important. I could just search for it. Cat star TSV, grab counting that particular thing once.
Oh,
Once it's
Once is all you need. We don't have any context on how many copies were in that whole thing. How many reads mapped to it, right? We started with a billion short, tiny reads. we wound up with tetracycline resistant protein class B is here. Is here is not super useful compared to is here in this percentage compared to this patient. So right now we just have kind of true false. We found these genes in this sample. So next up would be coverage script. This is what I want you running. I want you to run the annotation and coverage for next time. But coverage is doing the next step, which is we got our long contig here, and this gene was found on that contig. How do we determine how many times it was present in our sample? If I open this the GFF file, got a big header. I'm gonna cat the whole freaking thing. There we go. Ah, no. Okay. It tells us exactly what contig it was found on. Each contig has a name like this. How I annotated it, what type of gene it was, and then start and stop position. So it says this gene was found on this contig. Here's where it starts, here's where it stops, and then here's all the information about it. So if I cat, oops, the GFF file, search for tetracycline resistant protein class B, it's not found. I don't know why it should have. Okay, so tetracycline is probably just truncated. It's found 7 times. Let's just bring those up. It's because there's a little special character. It's found on node 3. It's found on node 3. It's on 27. It's on 63. Okay, so it's found sometimes, but we still only know that it was found on those particular contigs, not how often it was present in the sample. So what do? We got to go back to our quality control short reads. We need to map all of them onto our contigs, see how many times each one maps into each little spot here with misanoid. How many short reads mapped to this contig between these bases? That's going to give us essentially an estimate of how abundant that particular gene was in that sample. That, once we get that done, and I just tested the code that I had ChatGPT write for me, and it worked perfectly for the coverage. Once we get that done, we've got ourselves a file that resembles the TSV file. With a new column that's the number of copies per million in the sample. So it's like a normalized copy. It's kind of like a relative abundance. And then you're done. That's it. Bioinformatics over. Study's not over. The stats start, right? Then you import this tab separated nice file into, if you're a good person, R. If you're feeling crazy, then Python. And you do stats. I found this gene in this patient this many times, this gene in that patient this many times. How do they compare between our treatment groups? So once you've gotten the coverage done, congratulations, you are now an expert bioinformatician. And you pass it off to the stats people and say, here's the file. And they take this file that took you like weeks to make, and they throw it in the ggplot and make a volcano plot and say, look, answer. And they have no idea what it took. Get this. That's okay, keep the magic to yourself. Don't teach them how to do it, and be out of the job. But that's it. We should be able to have those run by next class. It was real short. Annotation took like 5 minutes on this sample. I know what yours picked, but asked for 10 hours just in case. And then the mapping took like 20 minutes. Those two can be easily done in the loop stage without having to make new Slurm jobs for everything. So get those, and then we're going to kind of look through it and kind of see the pipeline as a whole. And then on Tuesday, we'll assign the task, split up into groups, and start doing this for 10 samples so that I can import it into R and make pretty plots for you guys. Unless you guys want to do that part. It's good to play in R. You don't like R? You can do it in Python. Okay.
So for strict 4 and 5, do we need to know like the replacement things?
No, because they're like so fast.
Oh.
The assembly is the really computationally intensive long part. Mapping short reads to an assembled genome, this is what we kind of have. We have an assembled metagenome. That's so fast.
Oh,
So BBMap, Minimap2, all these different programs will do that real quick. It doesn't take much memory or anything. And it's just counting the number of times any read mapped to that section of the contig, keeping track of it.
So we're submitting the coverage and information you're going to go on this like the big thing.
Yeah, they need to be Slurm jobs, but you don't need to ask for a whole day or anything. A few hours to get it done for one sample. When you're doing your big project, a full day will be enough for 10 samples. No problem. I do like 100 at a time. For these like 2 steps.
yeah. Now how to the one for another?